Okay, to see if you understand transcription, here is a question for you. If the DNA molecule above were transcribed, which of the following RNAs might be produced? So you may want to pause this for a minute while you think about it. But what I want you to remember is that RNA molecules must be complementary and anti-parallel to the DNA template strand. Also, they must use uracil instead of thymine. So the correct answer is C and D. For right now, either of the DNA strands could be used as the template for transcription. So realistically, both of those are options of what could be produced through transcription. Alright, so let's take a more detailed look. So this is how we got to that point. So you see that DNA strand from the question before. Either strand can act as the template strand and can contain information for how to make the protein. So you can see the RNA strand, the complementary RNA strand that would be produced for either DNA strand using uracil instead of thymine. So that could have been the first thing that you would have done, was disregarded any of the options that had T's in them instead of U's. So the next stage of gene expression is translation. I want you to take a moment and think about the structure of a nucleotide and the structure of an amino acid. We've done these before. And what we are really talking about is how a nucleotide code can be converted into, so how an adenine, cytosine, guanine, thymine, uracil can be converted into an amino acid. There are only 4 nucleotides, but we have 20 different amino acids. So if a combination of 2 nucleotides corresponded to an amino acid, how many different amino acids could DNA code for? So the correct answer would be 16. Okay, so for a 2 nucleotide codon, we would have 4 squared. Okay, the number of different possible codons would equal the number of different nucleotides raised to the x power, where x is the length of the nucleotide string. So here are our nucleotides, and these would be those 16 options if we were using just a 2-letter code for an amino acid. But that's not what we actually use. We have a triplet code. So the bases in DNA and RNA form a triplet code to make an amino acid. So up on Blackboard is this codon table for you to use. You will probably want to save it or print it out so that you have it available for activities that we are going to do. And what you do is you look on the left-hand side for your first base letter, then you go along the middle for your second base, and then you go up and down again for your third base. What you will notice, as I go back for a minute, is that multiple codons code for the same nucleotide, and they tend to be very similar. So in translation, we have that messenger RNA which was created during transcription that is going to carry the nucleotide code for the protein to be made. Now we have another RNA getting involved, and that is the transfer RNA. These are the molecules that are going to interpret the nucleotide code. So each transfer RNA is going to carry an amino acid, on its 5' end, and it's going to carry a complementary anticodon on the other end that will match the codon in the messenger RNA. So this is an example of a transfer RNA that has an anticodon ACG that is complementary to the UGC on the messenger RNA, and it has an amino acid attached at the top, and that amino acid you can see depicted here in its structure. So during translation, we have ribosomes that are going to use those transfer RNAs to read the messenger RNA and assemble the amino acids that they carry into creating a new protein. So you're going to go and do another simulation activity. This time you're going to click on translation instead of transcription, watch the video, and then click on interactive and go through the steps of translation.